All right, let's get going. Do you want to get the slides moving and we'll start moving forward? There we go. Oh, that's me. How about that? Hi, everyone. Let's get started. I'm Will Alexander, dog show judge, educator, retired handler, and host of Dog Show Tips. And on behalf of Pro Plan and Paw Print Genetics by Neogen, I would like to welcome everyone to tonight into this webinar on small dog breeding, genetics, and reproduction. We have a great presentation in store for you, and I'm glad you could all make it tonight. First off, we have Dr. Westra, veterinarian, associate medical director for Neogen, joining us tonight. And I'm going to let Dr. Westra take over and let us tell us something about himself.
Oh, all right. Well, I'll do that in just a second when I start my presentation. That way you can see a little bit more about it. But here's just, those of you that are reading this can just kind of see a little bit about my background. It is a rather diverse background.
Graduating with a DVM from Washington State in 2008, Rob spent 12 years in a small animal general practice and emergency medicine. Prior to that, he capped off a 10-year career in wildlife biology, working with tropical fish, Pacific seabirds, and coral reef ecology. Rob enjoys the outdoors, the Pacific Northwest, providing a lap for his dogs to sleep on. And there we have Dr. Greer. We all know Dr. Greer. She's been on the show quite a few times. Dr. Greer is a renowned veterinarian and also a lawyer whose life work has been dedicated to improving the health and well-being of pets. She has been a Pembroke Welsh Corgi breeder since 1990, has extensive experience in breeding and raising show dogs, and has written a book called Canine Reproduction and Neonatology, a practical guide for veterinarians, veterinary staff, and breeders. She has also recently voted Westminster Veterinarian of the Year. And I know a few of you probably have voted for her. Yay, Dr. Greer.
So Dr. Westra is one of my favorite people to listen to when we go to seminars. He's amazing, great educator. I have yet to sit through an entire presentation of his. I usually either come in late or leave early or have someone tapping on my shoulder during the presentation. So I still don't understand everything he talks about because I have to listen to coat color and some of these things start to finish. But he's got such great material. You guys are going to have a blast tonight. This is so good.
It's going to be exciting. Our agenda for tonight will kick off with Dr. Westra's presentation on small dog breed genetics, a quick message from Paw Print Genetics by Neogen, followed by Dr. Greer's presentation on small dog reproduction considerations. Then a quick message from Pro Plan. We'll have a 5-minute break and we'll head right into a 30-minute of Q&A, wrapping up at around 9:30. We may go a bit longer tonight. And a little bit of housekeeping before I turn it over to Dr. Westra. I want to touch on a few things. We're a big group tonight, so if we're going to keep everyone on mute during the presentation, please enter your questions Please enter the questions for the Q&A period anytime in the Q&A box. It's a 30-minute Q&A period, and we'll get to as many questions as we possibly can. And please take a moment to note the difference between the Q&A box and the chat box. We'll be taking questions for our presenters exclusively from the Q&A box, but feel free to use the chat box to connect with each other during the presentation. Finally, the doctors would like to ask you that you keep your questions to general topics they've discussed today and specific to your own dogs. With that, I will turn it over to Dr. Westra.
All right. Thank you very much, Will. I feel very privileged to come and speak with you guys this evening. And I guess I'm a little excited because it's very, oftentimes I end up answering the same questions multiple times. And so this was an opportunity for me to kind of develop some new material to look at the specific genetics of small dogs. And so let me kind of get right into it. And I guess one of the things too is that I apologize that I'm of a generation that this format is kind of sometimes a challenge for me. So please apologize if I have technical difficulties. And at the same time, when I talk about genetics, a lot of times I have the ability to look at my audience and see where I'm losing them, see where they're glazing over, and I don't have that privilege here this evening. So if there's something that I say that doesn't make sense, or if you'd like me to repeat it or try to explain it using different terms, feel free to utilize the Q&A function, and we'll talk about it. And I'd be happy to go over whatever questions you may have. So yeah, just a little bit about myself. I know we talked a little bit about kind of some of my diverse background in the bio, and that I did come from a wildlife biology background because I got a master's in natural resource science, where the focus of my study was in the loss of genetic diversity in endangered species. And so that's kind of where my introduction to genetics came from. In that I was looking at what happens to alleles, which are sections of DNA, when we're talking about populations that are getting really, really right down to the wire. And then I moved directly over from my master's program into my DVM program at Washington State University. And then because of some particulars, basically I married somebody way smarter than me. And so I kind of hitched my wagon to her star. That meant one of us had to go out and actually earn a living. So I jumped into small animal practice and found that I had a much better fit in emergency medicine. And I just I did that for about 12 years before I had the opportunity to come back and do canine genetics. And that's why I started with a small startup company called Paw Print Genetics. Paw Print Genetics started in 2012. And then in 2021, it was acquired by Neogen. So Neogen actually started in 1982 in Lansing, Michigan, and Neogen started off as a food safety company. And as a result of working with food safety, that meant starting to work with agriculture and agricultural animals, and that eventually led into genomics. And I highlighted the last sentence there to kind of explain why Neogen is now doing canine genomics, is that because their focus is to allow customers to produce more efficiently and effectively than they could before. And that's one of the upsides of doing canine genetics, is that it really is there to help you as the breeder, not only to produce healthier animals, but help you to produce the animals that you're actually shooting for. So a little bit about what we're going to talk about tonight. We're going to talk about small dogs. I love small dogs. So that's just, this is where I said I'm actually quite Twitter-pated about this whole thing. And so to start off with, I want to just go through some of the just general genetics that we understand about what makes dogs small. And the focus of this is mainly just more or less to hopefully maybe increase your understanding of something, and hopefully it'll provide something that maybe you didn't know before. Some of you I know are certainly probably very conversant on these breeds, and so this may be either old hat for you or whatnot. But I'm hoping I'm able to provide something of interest to you. And then what I'd like to do is I'm going to actually go through a hard transition, and we're going to talk about a specific mutation which can affect the size of a dog. So it does have a direct application to tonight's topic, but it is quite a hard, or quite a discussion that has been going on since November of 2019, when a commercial genetic test became available. And then I'll finish off with looking at about what potential opportunities will exist in the future. So with that, let's get right into it. And we're going to talk about small dogs. And yes, I think we started off in the business, like what actually defines a small dog? And this is more than just short dogs, although they will be relevant to some of this conversation. But I kind of define it as anything less than 30 pounds. And what's interesting is that this trend is increasing within the United States, is that we have small dogs making up almost half of all pet dogs in the United States. And so this is quite a growing section of the dog population. Now, one thing that is interesting about dogs is that the domestic dog has the most variable size of any mammalian species on Earth, displaying nearly a 40-fold size difference between breeds. And then, I was out just cruising and looking for images of this, and I found this one, and this pretty much typifies the very distinct difference between breeds of dogs. And what is interesting about dogs is that these two breeds, could reproduce. We could get a Lab Wawa, and maybe some people actually have, which always creates some challenges. And I'm going to leave that to Dr. Greer's portion of the discussion. But that's what makes dogs interesting. Now, so when we're talking about size, we're going to be talking about morphometrics. And morphometrics very literally translates to the study of size and shapes. Now, and I'm going to talk about this concept multiple times throughout this presentation. That is, when we talk about morphometrics, we are dealing with a polygenetic condition. And that means it is controlled by many parts of the genome. Lots of genes are at work here. Different chromosomes are at play. And we're looking at not only protein synthesis, which is what genes typically code for, but also in the regulation and control of which genes actually contribute to the overall phenotype, or what we see when our dogs are produced. Now, as of now, and I can't say that I did a completely comprehensive lit search on this, but I went, I basically went around to all the resources, and I think we're looking at about 20 different genes that we are comfortable with saying have a significant effect on the size of the dog. Now, when we contrast that with humans, we're looking at height and size is controlled by thousands of genes. And so this is a little bit different in the sense that we're looking at, 20 for dogs, thousands for humans. And what's interesting about humans is the single gene that we've noticed is the greatest contributor, or the one that can produce the most significant effect, only accounts for about 0.5% of the overall change in size in a human. Now, I did put that asterisk in there because when we talk about human genomics, our understanding about human genetics is far, far greater than our understanding of canine genomics. So it's not that humans just have that many more genes, it's that we've just studied them for that much longer. And so we have a better understanding of how those genes interrelate. Now, bit of a review, but because I'm going to be throwing these words around, I like using this slide because it kind of brings it all back into perspective, all right? So we're talking about DNA, and where is DNA found? It's found in the nucleus of most of the animal cells out there. And then at very specific point in times in that cell life cycle, that DNA organizes itself into chromosomes. And chromosomes not only allow for cell reproduction, but it also becomes for the inheritability of genetic material to be passed from one generation to the next. Now, when you look at this diagram, you see that a chromosome is simply a coiled up section of DNA, which that if we break it all the way down to its base to its finite level, is we're looking at those base pairs. That's the A, G, A, C, T, G that you see at the end of the diagram here. That is the actual sequence. That's the code. That's what the animal can then process to either develop a protein or it determines a trait or what have you. And so when we're talking about chromosomes, dogs have 39 pairs of chromosomes, which I've always found is interesting because as we all understand, or mostly it's common knowledge, is that humans have 23 pairs of chromosomes. And so it's like we're looking at significantly more genetic material in dogs. Now, one thing is, does that mean dogs are genetically superior to humans? I'm going to leave that discussion for you guys, but truthfully, more DNA does not necessarily mean better. I had to throw in, since we're talking about genetically superior, I had to throw in this Hulkster next to us here just because I love this picture. But we understand that when we're talking about a lot of DNA, we understand that most of it is what we call non-coding. And that's going to become relevant to the conversation as we move forward. Just for your FYI, if you guys aren't aware, the creature that you're seeing before you is called a bully whippet, or is suffering from what we call bully whippet syndrome. And that's actually caused by a mutation in the MSTN gene, which limits the amount of myostatin, which inhibits muscle growth. So these poor kids do not basically have any inhibition of muscle growth. And so, of course, we're talking about size of dogs. So I had to put in this Hulkster here. Now, when we're talking about chromosome, we're talking about inheritance. So that means the passing of genetic material from one generation to the next. And it stands to reason that you get 50% of the genetic material from the father, 50% of the genetic material from the mother. It is this case always. And a gene, because we're going to use that term throughout this discussion, is the basic physical and functional unit of hereditary. Now, when we're talking about genes, one thing you know is that when it comes to humans, we know that insanity is inherited because you get it from your kids. So, it's one of those things where we need to relate inheritability back to our audience here. All right, so having looked at that, and we've talked about genetic material, one thing I'd like to talk about is what actually causes change in size in dogs. Now, we know, I said before that we're looking at about 20 different genes, but I kind of want to focus in on one in specific, and that is the insulin-like growth factor. What insulin-like growth factor is a gene that regulates the production of insulin-like growth factor. This insulin-like growth factor is produced in the liver, and it regulates other activities within the body that regulate size, such as just true growth hormones. And what makes this, or the reason why I picked this gene to talk about, is that this one gene is been accounted for about 15% of the dog size variance is just because of this one. Now, remember when we talked about humans, the big dog on the block there was one gene was responsible for 0.5%. But to our understanding to date, and I'm going to qualify it like that because at this point in time, we don't know what we don't know about genetics in terms of canine DNA. But so far, what we say is that this one gene, we are led to believe that it accounts for 15% of the size of a dog. So the IGF-1 hormone is a biochemical marker. We can actually, when this hormone's floating around in the blood, we can actually do a blood draw and we can measure its levels within any dog. We use this diagnostically in veterinary medicine, one, to account for some quantification of liver function because the hormone is produced in the liver. We use it diagnostically to determine if a dog is truly a dwarf. And we can also use it to verify nutritional status in a dog. So if there's been cases of neglect or a dog is not basically metabolizing the food that it's being fed, sometimes we can use IGF-1 levels to determine nutritional status. Now, to bring this back to the relevancy of tonight's topic, toy breeds have significantly less hormone than big dogs. Basically, small dogs have less IGF-1 hormone, big dogs have more. And one of the things we've become to realize is that the IGF-1 gene was long suspected to control this, but we didn't quite understand how. And what happened is then during the pandemic, during COVID, during the lockdown, a researcher was able to determine that there was a section of non-coding DNA. Remember, going back to those chromosomes, there's parts of the DNA that we don't think of as non-coding. Now, let me stop for a moment. When I talk about coding, we think of that DNA as the functional DNA. And when we say coding, it means it codes for protein synthesis, meaning that this gene causes the cell to produce a specific protein, which then has a job. And when we talk about the non-coding, we don't really understand what it is. We know that it doesn't produce a protein, but as our understanding of genetics is getting greater, we understand that maybe then this non-coding region of the DNA, we're finding out that this is where gene expression is controlled. This is where we find, and this is a perfect example of how sometimes a section of DNA doesn't actually produce a protein, but regulates how a protein is produced. And that's exactly what's going on here with the IGF-1 gene. So within this mutation was in the non-coding read, we determined that the IGF-1 was being controlled by that small dogs had a mutation that was preventing the production of IGF-1 versus big dogs. Now, I want to stop here and I want to talk about the concept of variable expression. Because when we're talking about size of dogs, I mean, we're looking at that 40-fold difference between small dogs and giant breeds. And so you think about that is that we have dog breeds that fall all along that spectrum from the very, very teacup Chihuahua all the way up to the Great Dane and the giant Mastiffs. And so when we talk about variable expression, we know that dogs can fit all the way along that growth or that size spectrum. And so we have to think about this is that when we talk about mutations like this, going back to that very first concept of polygenetic that I talked about, is that we have to think about this is that even though I'm talking about one specific gene, it's being controlled or regulated by so many other factors. Other genes, the other 85% that we don't completely understand. I talked about the 19 other mutations that we do recognize that control genes. And then there's this whole other half of it called the environmental component. And this is where nutrition comes into play, is that nutrition can have a big effect on either assisting, promoting, or actually inhibiting what is within the dog's basic genetic code. Oh, I've gone backwards. There we go. All right. Now, another interesting fact about the IGF-1 mutation is that they determined that this is actually an ancient mutation. This is the ancestral allele. In other words, this is what was found within dogs before, it basically, that dogs were predisposed to be small initially. But then over time, there was natural selection and dogs became larger. And that is due to a mutation within that gene. And then so when we start talking about dominant versus recessive, the mutation that happened that allowed dogs to increase in size was actually dominant to the ancestral allele, which we consider to be recessive. So when they did a DNA study, when they were looking for this mutation, they actually looked at several wolves in large domestic breeds, and they found this IGF-1 mutation, which should determine that the dog should be small. So you think about it, if a wolf is running around there with the recessive allele that codes for the inhibition of the IGF hormone, you would think, why are wolves small? And again, going to sound like a broken record here, but that's because this one specific gene that I'm talking about only counts for about 15% of what we understand to make up the dog's size. So there's that other 85%, which can allow a wolf to be essentially what would be considered a normal sized wolf, even though it has one copy of this gene that should basically limit it or cause it to be small. Now, two things happened in the history of dog genetics. One is that the dog domestication happened about 50,000 years ago. That's when they started, pick the story that you want to ascribe to. This is when dogs started showing up at the campfire and whatnot. But humans actually at that point in time started to select for smaller dogs because they found that these dogs actually had more of a functional role in their domestic environment. And this was that they started selecting for dogs for, such as rodent control, or the other one that was commonly touted was herding variety. They found that the smaller dog tend to be much more pliable to these activities than, kind of the more wilder monsters that was out there. So that happened a long, long time ago. But then because the mutation which codes for a large breed is dominant, there was a period of time where the allele frequency of the small gene was getting slower and lower. What happened then is that basically during the Victorian breed expansion, again, we started to artificially select for smaller dogs, and then we were able to kind of conserve that small gene, or that small mutation gene. And as a result, over the last 200 plus years, we have seen this resurgent and in fact an explosion of small dogs. Now, let's talk a little bit about genetic testing for size. Again, it's polygenetic. We know that there are multiple genes for it. There are 20 that we know of, but there's also probably, dozens, if not hundreds, that we don't know about. And they all contribute a varied amount. So because we know of these 20, we can develop commercial tests for them. In fact, there are some companies out there that do offer morphometrics or size determining tests. The problem with that is because it's polygenetic, because we have variable expression, we understand that getting a genetic test result that says your dog should be large, and at the same time you're looking at this little 8-pound dog, and you're like, okay, that doesn't make sense. One other challenge that we have when it comes to the genetics of size is that mapping of the traits is tricky, and there are many linked markers. And when we say linked markers, we mean sections of DNA which are very close to each other in proximity, which can confound our interpretation or exactly understanding what that specific sequence of DNA did to promote the trait that we are recognizing. So one of the known complications of size genetic tests is that we understand that there is a gene out there which causes ears to stand up. At the same time, we know that it is linked to a section of DNA which controls for small size. So when we find that, the question is do we have the sensitivity to detect that we're talking about the ears, or are we talking about the size? And so it could be very easy for us to say your dog has ears that stand up when they're clearly these floppy, wonderful little expressions. And at the same time is that we might say the dog is going to be small when actually it's just we found the gene that causes the ears to stand up. So we have to be very careful when we interpret these results. Now, some would argue that they still have a benefit to a breeding program, because if you get the genetic test results back that say your dog should be of a certain size, you can use that information to kind of increase the chances that you're going to get the size of dog that you're shooting for. But because there's so much variability in the actual presentation due to all of these factors that I've outlined here, you either have to do it with kind of careful concern, because a lot of times people get very, very frustrated that the results don't match up directly with what they observe within their dogs. All right, now I want to change gears a little bit, and I want to review inheritance just really quickly and talk a little bit about dominance versus recessive inheritance and then how they play into size. And then I want to talk about these two mutations, chondrodystrophy, which we shorten to CDDY, versus chondrodysplasia, which gets shortened to CDPA, and understand the difference of this and what this means for any breeding program. So first of all, to review inheritance a little bit, I've used the expression dominant and recessive. And so the definition of dominant is that only one copy is needed for that gene to affect the presentation into a dog. So when we talk about your test result or the genotype that you get from a genetic test would be something like capital E, where capital E is dominant. We would call that a homozygous dog. Or you could get a heterozygous result, which is where capital E and little e would be what you get in your genotype. But because capital E, which codes for the color black or dark pigment, is dominant to the light pigment, it doesn't matter if you get two copies of the same or just one copy with one copy of the recessive, you're still going to see the black phenotype. Contrast this with recessive, by definition, means that you have to have two copies of the gene inherited, one from the mother, one from the father. And in this case, in order for the recessive gene to be expressed or to see that in the actual dog, the genotype or the test result that you get is always going to be the two copies of the recessive, whether that's lowercase, or sometimes we'll call that a mutation and two copies of the mutation. But when you get two copies of that recessive, then that gene typically is observed in the phenotype. And in this example, I'm using the color yellow because yellow is recessive to dark or black. So if it's homozygous recessive, then you would see the yellow phenotype. Now, to relate this back to the genetics of size, the mutation that I talked about in the IGF-1 gene, the recessive was actually the small, the dominant was actually large. But the thing that confounds things here is that, because we have that presentation along a spectrum, that variable expression, if you will, we have to take into account that, again, there are multiple factors involved when we're looking at what actually determines the size of any specific dog. So one thing to understand, recessive does not mean hidden. Although most of the genetic diseases that we test for have recessive inheritance, we want to think of them as not as hidden, but basically as carriers. And this is how we use genetic testing to reduce disease within dogs. By finding out the carriers and then breeding appropriately, meaning that we do not breed carriers to other carriers or carriers to affected, we essentially can create or guarantee that we will not pass on the disease or the presentation of the disease by using the genetics of the dog. Because again, recessive requires two copies of the gene. One has to come from each parent. That opens up the door for this type of breeding. So if you know that your dog is a carrier for something, and you breed it to a dog that is clear, and in this example, the male is the carrier, the female is normal, in the offspring that's produced, the only thing we risk doing is producing more carriers, but we will not produce any affected or clinically sick animals. Now, I know there was a basset hound conversation, and I happened to throw one in there. I don't know how that falls. Do we consider basset hounds small, or do we consider them short? But I like them, so I put one in there. And so I want to use them to talk about dominant mutations, because dominant is something that a lot of people tend to kind of ignore. They just assume that, all mutations are recessive and I only have to find my carriers, and I don't, if I breed appropriately, everything's going to be fine. What becomes important is that if you're familiar with the mutations that affect your breed of dog, and that might require a little bit of, focused research on your part, but you must realize that there are some mutations out there which are dominant. And the ones that we're talking about tonight are just that. Both chondrodystrophy and chondrodysplasia are considered dominant mutations. And again, when the mutation is dominant, only one copy is required for the condition to be expressed. So an example of a dominant mutation, which affects one of my favorite toy breeds, is the Chinese crested. The difference between the dog on the left and the dog on the right is one copy of the FOXY3 mutation. And that's the difference between a powder puff crested and a hairless crested. Now, also what I love about the crested is it's another example of variable expression, because as people who breed cresteds know, the ideal was what they want to find is that quote unquote hairy hairless, which means that as you see with the dog on the right, there is some growth of hair on top of some wonderful environmental factors called grooming. But at the same time is the dogs that have the copy of the FOXY3 mutation will produce some hair. And the idea is that if they produce it significantly, if their coat is full, then it can be, modified into that, very stunning example that we see on the right. Another example of dominant mutations that most people are comfortable with is Merle. Merle is a dominant mutation. So if we breed a dog that is with one copy of Merle, or we'd say one copy affected, because there is no such thing as a carrier when it comes to dominant mutations, you breed it to the tricolor of the dog on the left, what you're looking at in the offspring is 50% normal or 50% non-Merle dogs, and 50% one copy affected or 50% Merle dogs. Think back to the same diagram I showed for recessive, where we had 50% normal, 50% carriers. It's a distinct difference now because we're dealing with a dominant mutation. Now we're dealing with dogs that have a 50% chance of producing or demonstrating the effect of that mutation. So that brings us to chondrodystrophy. What is chondrodystrophy? We're looking at a mutation called the FGF4 mutation on the chromosome 12. This mutation causes dogs to have an abnormal presentation of cartilage, and it only requires, because it is dominant, one copy of the CDDY for a dog to be affected with chondrodystrophy. Chondrodystrophy, by definition, is simply abnormal cartilage. This abnormal cartilage can affect the growth of the long bones. It can cause a shorter dog or a dog with a smaller leg. But more importantly is it tends to cause a weakness or a degenerative change within the cartilage that makes up the intervertebral disc, those chunks of cartilage that reside in between the individual vertebrae that make up the spine. When this happens, a dog is predisposed to intervertebral disc disease. Now, I've talked about variable expression when it comes to size. You have to understand that when it comes talking about chondrodystrophy, there is also variable expression of how affected that cartilage is. So how many discs are compromised, how short the dog would be as a result of this mutation. And in reality, what we have found, especially as we've started testing more and more of these dogs and talking with people about the phenotypic presentation of dogs with one or two copies of this, is that actually there is quite a bit of I guess what our concern is that dogs with this mutation tend not to have the same conformational changes as they would with chondrodysplasia, which we'll talk about in a minute. But what is more important is the abnormal changes that happen to the vertebrae. Now, what is intervertebral disc disease? Again, like I said, because of one or two copies of the CDDY mutation, it can cause this chunk of cartilage, as you see the diagram on the right there, this cartilage that resides, that basically acts like a nice shock absorber for the spine as we go along here, is that this becomes compromised. It effectively becomes more brittle and more sensitive to traumatic changes. Now, a lot of times people will get their test results saying, my dog has IVDD. I did the genetic test. That's kind of a misunderstanding of how it works. The genetic test is for chondrodystrophy. What actually causes IVDD is trauma. A dog with CDDY is therefore at risk of IVDD, but isn't necessarily, it isn't one of those things where having CDDY does not equate to having IVDD. So that's the concept of risk here, is that when we're talking about having CDDY, we're saying your dog is at risk or it's predisposed. And one thing to understand is that when we do a genetic test, we're talking about relative risk. That means what is the risk of your dog compared to all the other dogs that are clear of this mutation? So we're looking at your dog relative to that population of dog. When people call me on the phone, and what they're always asking about is, what is absolute risk? And that is, what is the likelihood that my dog with this mutation will actually present with the disease? And in veterinary medicine, that's a very difficult question to answer because we don't track dogs the same way we track humans. And so it's very difficult for us to basically develop a study in which we're going to look at dogs over the course of their life to say that dogs with this mutation have this % chance of actually presenting with IVDD. So we kind of are defaulting back to relative risk. And that puts things in a very interesting light, which especially for breeders, we have to be very careful of. Because when we're talking about chondrodystrophy, we're looking at something that is very significant within many populations. Now, I just pulled just some examples of toy breeds, and to show you, these are the % of dogs that we've tested through Paw Print Genetics that have at least one copy of the CDDY mutation. Miniature dachshunds, of course, 100%. We have not to this point found a dachshund that does not have at least one copy of chondrodystrophy. It's part of, basically, we would say it's factored into the breed. At the same time, what's a little interesting is that we see this very high frequency in toy poodles, in the English Cocker, and the French Bulldog. Now, with some variable frequency, we see it in the Shih Tzu and the Crested. And then what I found really interesting when I pulled this data is look at that Chihuahua. Only 7%. Now, granted, we haven't tested, thousands of Chihuahuas, but in the ones that we have, we're finding it in a very much lower frequency than we see with these other breeds. Now, when we're talking about genetic testing, 7% is still exceptionally high. We are often scared because we want all genetic mutations to be down below 1%. So 7% is still significant, but in contrast with these other breeds, I mean, that's a very shocking difference. Now, the point of this slide is that you have to factor this information into your breeding program, especially if you're working with poodles or dachshunds or Cocker Spaniels or French Bulldogs. If you're going to try to breed away from this mutation, you're going to go full scorched earth, meaning I'm only going to breed dogs that are free of this. When you're talking about French Bulldogs, are you really going to limit your breeding options to 3% of the population? That's the question, and that becomes difficult for breeders to kind of wrap their heads around, is that this is one of those situations because the mutation is found in such high frequency within so many of these breeds, and at the same time, because it is only it is a relative risk, meaning it does not mean the dog will get IVDD, does not mean the dog has IVDD. So it becomes something of a challenge for breeders to kind of incorporate this kind of testing. Now, let's contrast it with chondrodysplasia, the CDPA. CDPA is the FGF4 mutation, but this time it is found on chromosome 18. Now, in difference to the CDDY mutation, this one more consistently produces the phenotypic changes of short legs, but it does not cause the degenerative changes to the intervertebral discs. So what we find is that dogs with the CDPA mutation will have shorter limbs than dogs without it. Again, this is all variable expression and it's all relative within the dog population. But these dogs will have shorter limbs, but they will not be affected by something that we consider to be an abnormal pathology. So we've described CDPA as a trait, not a disease. In fact, it's the defining trait for the Dachshund and the Basset Hound, the Corgi, and many of your short-limbed breeds of dog. Now, pulling back up the 39 chromosomes that make the canine genotype just shows you that, and my arrow is off here, the CDY is found on chromosome 12. It should be moved up, my apologies. And the CDPA is found here on looks like everything's off. It should be found on chromosome 18, and that's up here at 12 versus 18. So we're looking at the same mutation, but found in completely different sections of the genome. And that partly explains the difference in the presentation that we see between these two mutations. So something that I get from a lot of breeders is that they're very excited about this dog that they found that's CDDY clear. And this is a picture of Mr. Happy Face. I think he was the winner of the ugliest dog competition back in 2022. But I use him as an example because not only is he adorable, but this is the kind of thing that you as the breeder have to step up and say, great, he's CDDY clear. Would I breed this dog? No. Because this dog probably doesn't help us achieve that kind of movement towards the breed standard, which is what we're doing when we're doing purebred dog breeding. And the other thing too is that you can't overlook dogs that may help you with your breeding priorities and the goals within your breeding your goals within your breeding program just because of one genetic test result. So I call that looking for the unicorns. That means is that I don't want you to overlook good dogs because of their CDDY status. At the same time, you don't want to place too much value on that one trait. However, having said that, I'm a veterinarian, so by nature, I can talk out of both sides of my mouth, is at the same time is that there is an opportunity for us to reduce this mutation within the frequency of our breed of dog. But the thing to keep in mind is that you're not going to do that within 1 to 2 generations. This is going to be something that over time, we're going to walk away from this mutation, which means that we find those dogs with one copy to breed to dogs with one copy, and that has a 25% chance of producing a clear dog, but it most likely will have a 50% chance of producing a dog with one copy as well. And then we have a 25% chance of producing a dog with two copies. But it's that percent chance that we might get a dog that's clear out of that, and eventually we can start breeding the one copy to clear. That increases our probability of getting more clear dogs to 50%. And then over time, we start breeding clear to clear, and then we'll get away from that. Now, because the mutation is dominant, do you see how it's much more difficult to get away from this mutation than if the mutation was recessive? Because with recessive, we can breed carriers to clear, and we won't produce affected dogs. But when you breed the dominant, even to clear, you're going to get some dogs that are going to be chondrodystrophic. So the challenge to the breeder is to address any potential loss of genetic diversity in a population, which would occur if dogs with this mutation are retired. I don't want you to retire 80, or I can't remember what my statistic was, but I don't want you to retire 97% of the French bulldogs out there just because of this mutation. In fact, if you are very specific and you keep overlooking dogs because of this one test result, you may end up creating more genetic problems than we would solve by getting rid of this mutation. This is proverbially throwing the baby out with the bathwater, which is something that we need to avoid. So having one or two copies of CVDY does not mean a dog has IVDD. It means a dog is at risk for IVDD. So that's something to keep in mind. And that at the same time, is given the prevalence of the mutation, we should not disqualify a dog from breeding program based on this alone. I always tell breeders, it's your job to look at the whole dog to determine if that dog has the features, traits, and qualities that you want to conserve in your breeding program. Now, one of the advantages is whenever we're dealing with a risk assessment is it's just that, it's a risk. So we can engage in preventative care to reduce that risk to any one given dog. And that becomes an opportunity to educate our puppy buyers on what that risk is and how they can minimize it. And just off the, just really simple, the one we can do for intervertebral disc disease is educate the importance of maintaining a healthy body condition and a healthy weight. Because all of that is going to increase the stress on the spine, which is ultimately what's going to cause one of those intervertebral discs to rupture or herniate, which would then be IVDD. So again, breeding dogs requires the balance. We have to conserve the positive features and traits that we want, and we have to balance that against the acceptable risks inherent in the breed. So last, I want to close on what is the future? And this kind of wraps it all up in the sense of this mutation that we understood about the IGF-1 mutation was just discovered in 2020, or was published in 2022. And that means that every year we are making new discoveries about how genes are expressed, where the mutations are within the canine genome, how we can detect them through genetic testing, and then how we interpret them, and how that becomes functional for the breeder out there who's trying to preserve or trying to produce a very specific dog. The other aspects of the future is that new technology is coming out every year. Our ability to either detect new mutations is increasing, and at the same time as we're having the ability to look at larger and larger sections of the canine genome and understand how that relates, because we're getting a much, much broader, deeper picture of genetics. And so that becomes really exciting about what's coming down the road here, hopefully within the next couple of years. And that means actual functional tests for other polygenetic traits, things that are important to you, like hip dysplasia or luxating patellas, or OCD of the shoulder, or any of those other polygenetic traits that affect the musculoskeletal system. But that's where we're going with this. And it really does become kind of exciting to know where genetics will have in the future. And last but not least, I just kind of leave on this, is that I always try to talk to my breeders about the industry because family matters. We're capitalizing on the human-animal bond that has moved the pet from the barnyard to the backyard to the bedroom, realizing that research shows that 56% of people report sleeping with their pet, and nearly 35% of those are children. And I'll tell you, yes, big dogs sometimes get on the bed too, but a small dog is going to find their way under your pillow no matter what. So it's just important that when we are breeding dogs, we have to recognize that these are going to be incorporated into families as family members. And so that really does motivate us to produce the healthiest dog we can. All right. I really appreciate you guys giving me the opportunity to speak, and I look forward to your questions.
Hi. I guess it's my turn.
Turn it over to Caitlin from Paw
Oh.
Print Genetics at this point.
I'm just going to give you guys a quick message on what Neogen offers and for genetic testing. We have Paw Print Genetics. So we offer the largest portfolio of canine disease, coat colors, and traits. So it's including over 350 different breed-specific tests. We also have breed panels, which are our breed-specific panels, which are a collection of tests that are commonly found in specific breeds. We offer parentage, so DNA testing that can establish the sire of a litter of puppies. We offer clear by parentage, which is an alternative to testing an entire litter. So allows you to test your puppies or to have clear puppies if your parents have been tested and are clear of those diseases as well. We also have DNA profiling, which is like a unique identifier for your dog. And we also do the single testing for any sort of disease-causing mutation, coat colors, and traits as well. We have comprehensive screening as well, which is our canine health check product, and that is over 320 disease-causing mutations, coat colors, and traits, all in one test. It's not breed-specific. And then we also do offer a comprehensive screening for cats as well. I know that that's not super relevant here, but it's called My Cat Scan, and it's 120 disease-causing mutations, coat colors, and traits for cats as well. If you guys have any questions on that, just give us an email at pbgaskus at neogen.com, or you can go to www.pawprintgenetics.com as well. So, all right. And next up is Dr. Greer's presentation, which everyone is really excited to hear.
All righty. So, little dogs, small dogs, they're so much fun, but there are some challenges. And of course, there are joys in breeding them. So we're going to go through about 5 different topics. We're going to talk about the nutrition prior to breeding and during the pregnancy. We're going to talk about how different they are in their breeding process compared to larger breed dogs. We'll talk about the appetite that they have during pregnancy. We're going to talk about whelping and the risks associated with that versus C-section, and then neonatal care. So we've got a lot to cover and not a lot of time to do it in. So we're going to kind of buzz through this. And as Dr. Westra said, we'll have Q&A at the end. So we'll have an opportunity to discuss some of the details of this as we go through. So first, I want to talk about nutrition during the pre-pregnancy period and during the pregnancy. It's really important that we have great nutrition. We're going to talk about raw and carb-free diets, calcium, folic acid, and DHA. So in general, you need to feed a well-balanced, great diet to maximize your success rate. I've been calling this feed the machine because unless we produce bitches with great nutrition and have our stud dogs on great nutrition and our puppies aren't going to come out with the ability to thrive. So we need to really manage their health, their nutrition, and their environment. So we need to feed the machine. And sometimes we get kind of lost in trendy diets and some other things. So I want to make sure that we're kind of getting back to the basics. Now, our recommendation is the Pro Plan diets that are available to you in the Canadian market. We have Sport 30/20 and we have the small breed puppy. And it's really easy to look at the macronutrients on a label of pet food. You can look at the fat, carbohydrates, and proteins really easily and see what the macronutrients are. But the micronutrients are really where things get into some detail. And it's hard to read the label. It's hard to compare those products one to another. And I can tell you from many, many, many conversations with clients that if they're having difficulty with bitches cycling or bitches not producing litters of the size that they expect, or the male dog's not producing adequate amounts of semen, that the first thing we look at is nutrition because it's so important. So we talk about what do you feed? What else do you feed? Because rarely do people feed just one diet. They usually somehow manage to dress up the food. And essentially what you're doing when you're altering the diet that's available commercially is you're taking a diet that Pro Plan or other manufacturers have spent millions of dollars developing, and then you muck it up by adding stuff to it that you really shouldn't be. And then I also ask what supplements they're on, because we can see a lot of complications with those. So my personal dogs eat dog food. They don't get a lot of other items put into their diet. If they do get something, they'll get some veggies every now and then, depending on what I've got in the kitchen, but they're generally on a dog food diet. The raw meat diets have a lot of issues associated with them. One is that they don't have enough carbohydrate in them. They're primarily a protein-based diet, and dogs do need carbohydrates to lactate, and dogs do need carbohydrates to make puppies. It frequently causes calcium imbalances. So if we're going to have a dog with a dystocia, with a difficult labor and delivery, it's often because the raw meat or the low carb diet doesn't have an adequate amount of calcium. And we don't want to supplement calcium during the pregnancy, but the calcium balance that's established by the pet food manufacturers is what your dog needs for normal growth and for normal labor, normal delivery, and normal lactation. The other concerns I have with raw meat diets are going to be that there's bacteria in there. There's salmonella, there's shigella, there's all kinds of really nasty bacteria in there. We do see parasites coming through. We can see neosporum and toxoplasmosis in those raw meat diets. There was a litter of puppies that was produced that there was a pathology report presented to us at one of the meetings that I went to for puppies that ended up in neosporum. It was a litter of Doberman puppies that the entire litter was born with some serious health problems and all passed away by 6 months of age. Unfortunately, because it was laboratory information, they weren't allowed to share that information with me, but it was a pretty horrific situation. And then I think we see an increased risk of pyometra in dogs that are on raw meat diets, again, because of the bacteria. So we want to be really thoughtful about that. The calcium imbalances after a bitch has her litter of puppies can lead to some pretty significant things. This is a bitch with eclampsia that presented at our practice. She was a little dog. She had 7 puppies that she was trying to nurse. She wasn't eating particularly well. You can see that she's pretty slender. And the reason that she's in this position is because she's basically looking like she's having a seizure. But this is what eclampsia looks like in the dog. When these small breed dogs have large litters that are heavily demanding the calcium being immobilized from their bones. So we can see these kinds of issues with diets that aren't adequate. So she came in looking like she was seizing. I've seen this multiple times over the time that I've been in practice. And it's actually not a seizure, but it's calcium deficiency causing this muscle tetany. In cows, calcium imbalances cause the cows to become muscle weakness, and they go down. Dogs have the opposite effect. They have this rigidity and basically a seizure type of activity. And if we don't adequately correct the calcium balance, these dogs will die. Like I said before, we need carbohydrates to make puppies grow in the uterus. We need carbohydrates for these bitches to lactate. So it's important that we're making sure that dogs get carbs. So why are we feeding a no-carb diet? I'm not really sure where this whole trend came from, but dogs need carbohydrates in their diet. I know people say, well, dogs don't eat carbohydrates, or dogs evolutionarily didn't eat carbohydrates. And in reality, they did, because when they ate the critters that they caught, whether they were rabbits or other little creatures, they ate the GI tract first out of those animals, and then they would eat the rest of the animal, the rabbit, the muscle, the skeleton, that kind of thing. So they were getting carbohydrates in their diet. In fact, that's what they go for first, is the GI tract. So without carbs, we're going to end up with problems. So that's the pea-based diets can cause us some real problems. I had a client in the other day that was feeding a diet that was not a Purina diet. And I was looking at the labels. She had told us what she was feeding, and the dog was having some health problems not related to breeding. But I looked at the diet, the label, and there were 4 places that peas were listed on the diet. One was pea fiber, one was pea something else, one was pea protein. They had it listed in different ways so that it kind of hid the fact that pea was the number one product in the diet. So even though it looked like fish was the number one product, it was actually peas because of the way they buried it in the label. So you have to be pretty suspicious for any pet food label that's got those kinds of things listed on it. So we've seen potato-based diets and pea-based diets, a lot of odd kind of diets over the last few years that are causing us a lot of problems. How do I feel about raw meat? Well, I think eating raw meat is sort of like this kid kissing a pig. I think the bacteria, the parasites, the the stuff that comes from a raw meat diet very similarly makes me think of this. So the next time you want to feed your dog an exclusively raw meat diet, please think about it. And if you must do it, at least please don't do it during pregnancy and lactation. Okay, the second thing we need to talk about nutritionally is folic acid. And we know folic acid is in many of the pet foods, but it may not be in adequate quantities for our breeding animals. Again, it's a micronutrient that's hard to read on the label. So there was a study done in Poland in 2013 that showed in pugs and chihuahuas that with folic acid supplementation, they could reduce the incidence of cleft palates and other midline defects, including cleft lips, by half to 66%. So this is pretty compelling evidence that some of our small breed dogs are definitely in need of a folic acid supplement, even when it looks like there's enough in the diet. If we have dogs that are genetically predisposed to having this, and I know there's been work looking for the cleft palate genetics, but to my knowledge, the samples that we used to send to one of the diagnostic labs trying to find out the DNA marker for it, to my knowledge, has not been determined yet. I'm sure Dr. Westra is better informed on this than I am, but if we have breeds like the pug, the chihuahua, and the other brachycephalics, we can really significantly reduce the incidence of cleft palates with that. Now, I'm going to show you a series of pictures that are not really pretty. So if you are sensitive about those things, this might be a time to not look at your monitor for 5 or 6 slides. But I'm going to show you a series of pictures of dogs that we've seen in our practice that have midline defects. The one most people are familiar with is going to be cleft palates, but there are also cleft nostrils, open abdominal wall, open thoracic wall, spina bifida, and hypospadia. So cleft palates and cleft lips look like this. This is a yellow lab puppy on the left, so not a small breed. On the right-hand side is a brachycephalic breed, so you can see left in the roof of the mouth of the puppy on the left, and then you can see that there's a cleft palate on the puppy on the right, along with 2 cleft nostrils. We see a lot of cleft nostrils and a lot of cleft palates. They're pretty common in our dogs, much more common than we'd like for them to be. This little puppy that I showed you in this picture also had the misfortune of having spina bifida. So that red dot between her shoulders is where her spinal cord is open. And then between her back feet are her intestines. Off to her left is her placenta, but between her back feet are her intestines. So this puppy had the 3 defects of midline defects. So she had the trifecta. Fortunately, out of a litter of 10, she was the only one affected. If there is a situation where the whole litter is affected, that's often environmental, like a drug that was given inappropriately during the pregnancy. We tend to not see genetics cause that high a percentage of 100% of the puppies affected, but we can certainly see individual puppies affected by midline defects. There are 2 kinds of open abdominal walls, and I want to distinguish between them because we see these again fairly often. Had one last week. There's one that's called an omphaceal, and that's right on the midline. So that's right where the umbilicus is, that the intestines are exposed because the abdominal wall is not complete. And in that case, the intestines are covered with a peritoneum, which is the lining of the abdominal cavity. So it's going to have sort of a saran wrap type of covering over it. Those puppies we can sometimes save if they're born by C-section or if they're born at home and kept clean long enough to get to us. Sometimes we can open the abdomen and get those abdominal contents put back in and save those puppies. I'd say maybe half the time we can save them. The other defect is off the midline. So it's usually to the right of the umbilicus, and it's called gastroschisis. Now, I've talked to one of my clients who's a pediatric surgeon, and I've done some looking at this, and that is most frequently on humans because we can see these both on the human side. Most frequently, it's seen in teenage mothers, so young mothers who are Caucasian, who smoke, drink, and use other illicit substances. I'm guessing most of you don't let your dogs smoke or drink, that they're probably not Caucasian, although they may be teenage mothers. They usually don't have the peritoneal covering over them. And what happens with these is it's off the midline. And usually these puppies have an incomplete intestinal tract. So somewhere along the intestines between the stomach and the rectum, the intestines have a blind pouch so that they aren't complete. So even if you try to correct these surgically, these puppies don't survive, or they may not have an open rectum. So it's important to distinguish between those when you're talking about what the defect happens to be, because there are differences in the genetics behind those. So these are two puppies born with open abdominal walls. The puppy on the left also was a puppy with anasarcus, so it's not normal. You can see the distension of its feet and the belly. And then the puppy on the right had a pretty significant defect. This is not a puppy that we can fix because of the severity of the problem. This is a puppy that was brought to our practice last week. She was born at home with her intestines out, and the intestines were not complete, so we did not attempt to try and save this puppy surgically. Rarely we will see an open thoracic wall or the entire abdominal cavity open along up into the chest. Again, these puppies aren't puppies we can save because of the severity of the exposed tissue and the fact that if you did try to put some of this back, these puppies will sometimes be born with beating hearts, and so it can be pretty gruesome to see these. But if you tried to put all that back into the puppy, there's not enough puppy to go around. So we can't correct this because there's not enough abdominal or thoracic wall to close over this. So we lose these puppies, and it's pretty rough to see these born at home. The other defect that we can see, and it's not uncommon, is called hypospadia, and that's where the urethral covering over the penis on the male dog is open on the midline. Many of these puppies can do well. They can end up having a surgical correction. So this is called hypospadia. I had a picture of this up on my computer a couple of years ago, and my staff, I had 3 staff members walk by, and one of them said, "Ooh, who's got the hypospadia?" And the other one said, "Oh, hypospadia, huh? Look at that." The third one said, "Wow, are we going to fix that hypospadia?" The picture was sent to me by a veterinarian who didn't know what it was, but my whole staff identified what the defect was. So it's not uncommon, but it's not common either. Some of these puppies do fine even without any surgical intervention. But again, these are all midline defects. So anything down the middle that doesn't close correctly is a midline defect. So are soft spots or open fontanelle. And it's always interesting to me when people are horrified that this stuff happens. And on the other hand, I think of it and say, how is it that when your body is so complex that you started off as 1 cell, then 2 cells, and then you grew into an entire organism, that the right side and the left side finds each other as accurately and as many times as it does? So it's not how does this happen? Why does this happen often? The answer is, why doesn't it happen more often? It's sort of a miracle to me that we don't see more of these. Next is DHA. DHA is a fatty acid. There has been pretty extensive research showing the importance of this in the diet of bitches that are being used for breeding. So it is recommended that DHA is included in the diet. Purina does include that in their Pro Plan diets. So it's important in brain and eye development. We know that DHA added to the puppy formula does improve how smart our puppies are. There was a study done that indicated this, and so it started to be added then also to human baby formula, which I think is really fascinating that the veterinary side supported the research on the human side. CCI is Canine Companions for Independence. They raise service dogs. They're a 35-year-old organization that raises service dogs mostly for mobility, for people that have deficiencies in their mobility, that these dogs can help them in their life. It was discovered after looking at 200 different nutrients that DHA was depleted in bitches that had multiple litters. So litter 1 of these bitches, when they had their first litter of puppies, 50% of their puppies were graduating from the CCI program. And by the 5th litter, they were down to a 25% graduation rate. So the research was done looking at 200 different nutrients, determined that it was DHA. So DHA is now added to not all dog food, but it is added to the Pro Plan diets. So make sure you're feeding a diet that does have the DHA added to it in adequate amounts. Smarter puppies are always better. There was research also published in 2012 that indicated that DHA did not cause any kind of negative effects, but it did improve cognitive skills, memory, psychomotor function, immunology, and retinal functions, eye function in growing dogs. So great nutrient to be paying attention to. So that's nutrition. Number 2 is going to be the mating. And the differences that we see in small breed dogs are typically shyness, size differences, and the AI pipette size that you may be using. So most large breed dogs are pretty bold when they come to the veterinary clinic. They're pretty self-confident. We have a lot of dogs that come into the practice, especially the boys that know where the collection room is. So they march right in the front door and right down the hall, and they don't even have to have somebody on the leash. They know exactly where the semen collection room is because they look forward to those trips. But some of the smaller dogs, especially if they haven't been handled a lot, tend to be a little bit shy. They can be a little bit hard hard to convince that we should be okay collecting them. Some will have white coat syndrome, meaning that when we walk in the room with our lab coats on, that can be a little bit intimidating to them. So some of these dogs need a little bit of practice to figure out that we're going to collect semen and that semen collections aren't a bad thing. We do see size differences. Clearly, this isn't the kind of breeding that most of us are looking forward to doing, but we can see size differences, and that can make some of these smaller breeds difficult to manage their breedings, because some of the males might be on the smaller side and simply not capable of doing an adequate job of mating a female. So we may have to stand him on some kind of a support. We may have to put her on a lower surface, or we can do AIs, artificial inseminations. Most AI pipettes are around 6 to 8 inches long, but Revival has developed this smaller one that's about 3.5 inches long. It's really flexible. So it is a really nice, tiny little size. It's really only a couple of inches long. So for those of you doing your own artificial inseminations, if you're struggling with the larger pipettes that they seem just a little too rigid or a little bit too long, it's easy enough to order those smaller pipettes and get your AIs done adequately. Third, during the pregnancy, we see a lot of times that we have pretty fussy eaters, and there's 3 times during the pregnancy or during the heat cycle that we see this happen. First is during the heat cycle, a lot of bitches just don't eat well. They're in heat, they're distracted, they're thinking about boys, they're thinking about other things. So many bitches don't eat particularly well during their heat cycle. The second time that we see a loss of appetite is between the 3rd and 5th week after breeding. That is known to be the most reliable indicator of pregnancy in the bitch, other than ultrasound. Of course, ultrasound is always the best, but a lot of bitches will either slow down how fast they eat their meal. They may take 20 minutes instead of 20 seconds. They may not eat their meal at all for several days, and that's a really reliable indicator of a pregnancy. So not uncommon. We don't worry about it too much, except when we get to the extreme of them simply not eating anything at all. And then, of course, the day or two before they go into labor, many of them drop off on their appetite. So what do we need to do about that? And the smaller dogs are always the dogs that are most difficult to get them to eat, because they are already just a little bit of a fussier eater than our large breed dogs. You can put anything down in front of a Labrador or a Golden Retriever and it'll go down. But our small breed dogs are a little bit fussier and a little bit harder to get to eat. So you can use a variety of different foods. You can top dress her food with some of the Pro Plan canned food that will really help. B Strong is one of the B complex vitamins. We know B complex vitamins can improve appetite. So there's some things that we can do to really improve her appetite and make sure that she's getting the right food. Sometimes just changing the variety, the flavor of the food that you're feeding can also help. So there are some things that you can do to keep her appetite good, but top dressing the food can really help. And we know that the Pro Plan small breed, or the puppy small breed puppy food, has a more nutrient dense capacity, so that it's going to be fewer amounts of kibble or fewer amounts of bites of canned food will get more calories and more protein and more nutrients into her. So those more nutrient dense foods can be really helpful when you're struggling. And I think the bigger the litter, the less well a lot of bitches eat. So that's the time you feel like, okay, this is when you really need to eat and you're not eating anything at all. It can be really upsetting to people. So have some of these cans of food around and ready to go. Fourth is our whelping, and we can certainly see problems with whelping when we have these smaller dogs, especially. We can see that there's a mismatch in the size of the bitch to the size of her puppies. I would love there to be a study. So if anybody knows a nice veterinary student that wants to do some research on this, I would love for somebody to be able to take an x-ray, measure the size of the puppy's skull, measure the size of the bitch's pelvis or some other structural part of the bitch that you can see on an x-ray, and be able to say, this puppy's going to weigh this many ounces, so this is what you've got. Because if you know you have a Papillon and she's going to have a 2-ounce puppy, you're going to be like, well, I think she can probably deliver that. But if somebody says to you, she's going to have a 6-ounce puppy, you're likely going to say, whoa, I think I need to schedule a C-section. So I think we need some additional research on this. We don't really have a way to look at the x-ray and compare the size of the puppy's skull or body to the size of the pelvis. There were some studies done in French bulldogs, and they did some really complicated measurements of the pelvis and angles and all kinds of stuff. We need something really much simpler than that. So we don't currently have that tool. It'd be great to have. The other thing that, of course, becomes a problem is litter size. So sometimes we'll have small bitches with really large litters, and there's an upside and a downside to that. So this is a little bitch that came to our practice. She had her puppies, all of her puppies but one over the weekend, and came to us on a Monday because she'd been to the emergency clinic. She hadn't had this last puppy, and they told her that they could do the surgery, but she needed to pay them right away, and she didn't have the money. So she came into our practice because she had called 7 other veterinary clinics and no one would help her. When I opened up this dog's abdomen to take out the remaining puppy, there was green fluid already free in her abdomen. Her kidneys didn't look good. Her glucose wasn't good. She was septic. Fortunately, we saved her. We were able to take her to surgery, correct the problem. This was an accidental breeding, one that she wasn't really planning on, but this puppy clearly wasn't going to come out. Because of the mismatch in size, the puppy was just too big for her to deliver. So again, kind of a nasty looking picture, but it's the reality of what happens in real life. So is a litter that's a bigger litter better, or is a litter that's a smaller litter better? And that's a really difficult question. The bigger the litter, the smaller the individual puppies in most cases, which means that it can be easier for her to deliver the puppies because they will fit through her pelvis better. But once you get to those smaller puppies, they can be more difficult to raise because they are smaller, because they are more fragile. So it can take additional work to raise those. So do you really want 9 or 10 French bulldogs in a litter? And we've seen that. Or do you want one giant French bulldog? What exactly is the ideal situation? And the answer is, you don't get to pick. She gets pregnant with the number of dogs that you have, so you don't get to pick. But once it's decided, you need to figure out how you're going to manage the problem. So this is a picture of a litter of Cavalier King Charles Spaniels. This was a planned C-section. She had, yes, there are 10 of them. I had a litter of 10 Corgi puppies. When you get these small breed dogs with really large litters, it's a challenge. Anything this large for a litter, no matter the size of the dog, anything over 9 puppies, we recommend a C-section. When we took this bitch's x-ray, her owner could not wait for us to get this dog scheduled for a C-section. She's like, no, we are not going to try and free up a litter of 10 puppies. I'm not going to be successful. And by the way, she raised all 10 of them. She was very successful with this. So how do we schedule our C-sections? We've talked about this in prior presentations, so I'm not going to go into all the detail of it. You can go back and pull up some of the previous Purina presentations that we've done. But with good progesterone timing at the time of the breeding, we can schedule C-sections very effectively and have safe C-sections during the daytime, so you're not in an emergency situation in the boonies in the middle of the night with no help. So I put this picture of a push me pull you up here for those of you who are too young to identify a push me pull you with Dr. Doolittle. That's what this is. And I want you to do this because I want you to think about not just using progesterone at the beginning of the pregnancy to get her pregnant, but also at the end of the pregnancy to get her unpregnant. Because if we have good timing at the beginning of the pregnancy, we can very effectively put you on our surgical schedule, know what day she's due to have her puppies, and get those puppies delivered without ending up with a middle of the night crisis. So please, 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 I know you don't always need progesterones to get your females pregnant, but please do your veterinarians a favor and do yourselves a favor and have the timing done so that you don't get caught with a difficult situation. So we know bitches are pregnant 63 days from ovulation. They can have their C-sections as early as 61 to 62 days after ovulation. So if you have that information, you can get on your veterinarian's schedule. So pregnancy is 63 days long from ovulation, 65 days from the LH peak. So you can use those 2 tools as a way to time your C-sections. What else do we need to do? Well, puppy count x-rays are absolutely essential. I can't tell you how much cannot overemphasize the importance of a puppy count x-ray. It's really a good idea to know how many puppies are there so that you know when you're done, you know if you have too big a litter, too small a litter, if the puppies just look like they're too large. This gives you a lot of insight. And there are some people who have had concerns about their veterinarians not getting adequate x-rays. So there's a couple of tricks. We've talked about these before, but just to reiterate, number 1, take her to the veterinary clinic fasting, preferably in the morning, so that she goes in with an empty stomach. It's a lot easier for us to count puppies on the x-ray effectively without a big amount of food in the stomach. Number 2 is if she has stool in her colon, you can put in paper matches or a suppository. Paper matches are easy. They're easy to find at the store. You can match her, take her outside, walk her and allow her to have a stool so that those puppies are a lot more visible on the x-ray. And then I take both a right and a left lateral. It doesn't matter if it's right and left or left and right. Either way, you want to take 2 views, one with each side down. Encourage your veterinarian to work with you on this so that you can get accurate x-rays. It's going to save you a lot of grief. It's going to save your veterinary clinic a lot of grief. So be sure you're working with your vet clinic and your vet technicians to learn to do these x-rays adequately. And they should be taken after day 55 of pregnancy. Presumably, most of you already have digital x-rays in the practices that you go to. As Dr. Westra indicated earlier, we see these huge differences in the size of dogs. No other species on the planet has this size difference. What I really am baffled by are Shelties. Like Shelties can be 9 pounds or they can be 60 pounds. I'm baffled that a breed can have a 6-fold difference, but we can certainly see that big of a difference in our breeds of dogs. So knowing that information is really helpful. And we know our birth weight puppies often will be as little as 100 grams when our larger breed dogs may be 700 to 900 grams each at birth. So it does make a big difference. This is a nice illustration. This is a friend of mine with corgis. I saw a couple corgi people on the call that had 2 corgis, bitches, bred the same day, whelping the same day. She came over to us for a C-section, and she had a litter of 4 and a litter of 8. They are the same size dots on the incubator mat, so you can kind of measure how big those puppies are. But the litter of 4, you can see that the puppies were twice the size as the litter of the puppies that were 8 in the litter. So remember that you're going to get these small for gestational age puppies in some of your litters, especially the small breeds. So the larger the litter, the smaller the puppies, the smaller the bitch, the smaller the puppies. And when we have these smaller puppies, they're harder to raise. They're just more difficult. So we know low birth weight puppies, according to the Neogen research, have an 81% greater chance of death in the first 48 hours. And puppies that are the smallest 25% of their breed have an increased risk of mortality in 48 hours. So those are really important things. And the reason the tree is on this picture is it kind of reminds us that the placenta is the source of all food and nutrients to these puppies before they're born. And if there's so many puppies in the litter that you have 10, there's just not as much real estate in the uterus as there would be if there are smaller litters. So without the larger amount of space for the placenta to attach to, you have smaller puppies. We also used to say that puppies could lose up to 10% of their body weight, and it would be okay. But the research from Neogen, which is the vet school in the south of France in Toulouse, indicates that if we have a 4% or greater weight loss, that puppies have an 8-fold increased risk of death. So we need to give these little puppies a lot more support than we do if they're bigger, robust puppies at birth. It's also important that we get great lactation. Again, it's really important that these puppies are small, that they get enough to nurse. We're going to talk about supplemental feeding, but if they're not getting supplemental feeding and you've got to count on them to nurse, then we want to make sure that our bitches get started nursing right away. The picture on the right is a little Boston Terrier that had a large litter of puppies. She was nursing her puppies on my surgery table before she left our practice so that they all got colostrum and all got off to a good start. The picture on the left is one of my farm dogs nursing her puppies, and you can see that she's adequately nursing as well. You can see the Boston Terrier has enough milk. If you don't have adequate amounts of milk, metoclopramide is a prescription drug that your veterinarian can prescribe for you. The dose is 0.3 milligrams per kilogram every 8 hours. So talk to your veterinarian if you're having difficulty with your bitches not lactating effectively, getting that medication prior to the time that you're in trouble. The other things that we've talked about before, our bitches need carbohydrates to lactate. So you can do things like root vegetables as a supplement. Sweet potatoes are one example of that. I'm not sure why bratwurst help bitches to lactate. I have never quite figured it out. There's not fenugreek in those recipes. So I'm not sure why bratwurst work, but honest to God, it does. And then the other thing human lactation nurses recommend besides sweet potatoes are oatmeal. So these are some things that you can feed your bitches if they're not adequately lactating to help bring in their milk. Neonatal care is going to be our last topic for tonight, and that is going to be involving environmental support, nutritional support, and our smaller puppies simply just need more help. So environmental support is really essential. We know that puppies need plenty of oxygen. They need to be kept warm. They need to be kept hydrated, and they need to be kept fed. And like this box you can see on the right that's crushed, if one side of this cycle goes off, the whole thing goes off. So if we have puppies that don't get enough oxygen, puppies that get chilled, puppies that don't get enough nutrition, everything goes off the rails and we start to lose puppies. So we need to give our puppies heat sources. We need to give them oxygen in some cases. There are great oxygen concentrators you can purchase now to have at home. It's important that we keep them well hydrated and well fed so that we keep these puppies going, because if one side of this fails, the whole thing fails. So important to give them only good choices. This is a whelping box that I had for my litter of 10 Corgi puppies. I've had the same whelping box for 25 years. Works great. I love heat support from underneath, not from above. So these are heated whelping nests. On the right-hand side is a heated whelping nest as well. It's partially covered by a mat because the puppies are a little bit older. They're up walking, so they don't need quite as much heat. But when they're really small, they do need that. And we want to give them a place that they can only make good choices. So you can see that the puppies can move around so that they're not always on the heated nest, because if they're too warm, they want to be able to move off. But I don't want them to be able to wander so far away that they get chilled. So we only want to give our puppies good choices. So they need to be in a whelping box or some kind of an area that they're confined and it can't go wrong. So I want to just show you the next picture because I think this is kind of an interesting photograph. The picture on the right is a litter in a commercial kennel, and this is how they manage their commercial kennel. They have 33 heated whelping nests with geothermal heat in an Amish community where they have the ability to manage these whelping nests right down to the 10th of a digit of heat with a central command station. They don't have electricity. They use geothermal heat. So even in these kinds of settings, you can have really great opportunities to raise your puppies. So this is a whelping nest at my house. This is a whelping nest at a commercial kennel. There are many ways to do this, but heating from underneath is always your safest. Heating from above with heat lamps has risks associated with it. I don't recommend that. Again, there are plenty of places to get oxygen concentrators, and we have this nice incubator that Puppy Warmer makes. They are a great combination of products that you can provide oxygen and heat for your puppies and keep them well oxygenated, warm. And then, of course, the hydration and the hypoglycemia are up to you based on feeding these puppies. So if they're not getting enough to nurse, we can supplemental feed. So do you need to tube feed your puppies? Well, some puppies need to be tube fed and some do not. This is a great example of a graph for a client that was weighing his puppies, had them identified by color, and he could see that his puppies, some of them are gaining better than others. So he was able to intervene with supplemental feeding. Do you need to supplementally feed? Well, if they're not gaining weight, and they should gain weight every single day, if they're losing more than 4% of their body weight, they need to be supplementally fed. If their urine isn't pale yellow, they need supplemental feeding. And puppies should double their birth weight in the first 7 to 10 days after birth. So if those things aren't happening, the answer is yes, you do need to supplementally feed. There are 2 ways to do it besides nursing on the bitch. One is with bottles. And there are nice bottles on the market, nice nipples on the market. Miracle Nipple makes one that comes attached to a syringe. There are 3 sizes for those. On the left-hand side of the nipples that Puppy Warmer is developing, that'll be a silicone nipple, that it's a really nice soft nipple. The bottle on the right is made by a company in Germany called Newbie. It's really meant to be a medication dispenser for small children. But again, you can use it for larger breed puppies. And then the nipples on the bottom are kind of unique in that they are silicone, similar to what Ken is making at Puppy Warmer. They're not food grade silicone, but they do make this nipple style that has a shield on it if you have a cleft palate puppy, so you can bottle feed instead of tube feeding. Tube feeding is a very important skill for people to develop, especially if they have small breed puppies. This is a client feeding her puppy with a Miracle Nipple at our practice. You can't see the puppy's head as well as I'd like, but it is visible. I'm helping her feed this puppy, and she's got the syringe in her hand and is slowly syringing the formula in. What I'm encouraging her to do is kind of stimulate the puppy, and I want that puppy to really grab onto the nipple, wrap its tongue around the nipple in a C shape, and really suck on that. I don't want her to just shove the formula in because that puppy may aspirate it. So we want to make sure that the puppy has got that suction on the syringe so that it's really working the way that the Miracle Nipple is intended to work. So we can feed with a nipple or we can tube feed. And a lot of people get really anxious about tube feeding when I speak and ask people about. Half the people in the room say, I am absolutely confident in tube feeding. And the other half say, I'm terrified of it. So we're going to go through tube feeding. We've done this before, but I think it's so important that we're going to do it again. There are 5 P's of successfully tube feeding your puppies. So we're going to go through the 5. One is to pre-measure the tube from the tip of the nose to the last rib so that we know that we're in the stomach. If the tube stops short of that, you may be in the trachea. So you don't want to tube feed if the tube didn't go in all the way to the mark that you put on it by pre-measuring from the tip of the nose to the last rib. Number 2 is you want the puppy and the formula warm, because if we feed a puppy that's chilled, they will develop ileus and aspiration pneumonia. So you don't want to feed a chilled puppy. Number 3 is you pass with the chin down. A lot of people want to tip the head up and look at where the tube is going, but that's going to open up the airway. So you want to keep the chin down. You want to pass to the left. And the last thing you do before you tube feed is you want to pinch the tail or the toes of the puppy so you can hear it cry. And when it's crying, you know that you're not in the trachea, because a puppy can't make noise if the tube is in the trachea. It can only make noise if you're in the esophagus. So make sure that you pinch before you feed. And remember, even though occasionally you may have an accident during tube feeding, more puppies will die of starvation than will die from accidents from tube feeding. So you need to be brave and remember that you can do this. This is a video of us feeding a Rottweiler puppy.
Lay him on his side.
So this is a little puppy that came to the practice that was with a litter mate of 1, and the owner was concerned that he wasn't gaining the way that his brother was. He didn't seem to be thriving. So she came over to us and we taught her how to tube feed. I measured the tube, had a mark on there. I'm going to pass, you can see, with the chin down to the left. And then I'm going to hold with my left hand, because I'm right-handed, the tube in my left hand so that as the puppy moves his little jaw and moves his little feet, that he doesn't push the tube out. The formula quantity to be feeding is 1 cc per 1 ounce of body weight, or 30 grams of body weight. So it's an easy 1-to-1 relationship if you're measuring in ounces. If you're in Canada and you're measuring in grams, then you want to do that conversion. So it's 1 cc per 1 ounce of body weight. So a 4-ounce puppy gets 4 cc's, a 2-ounce puppy gets 2 cc's. It's pretty simple, straightforward math, nothing complicated at 2:00 in the morning. So you want to measure the tube, pass the tube, and then you want to go ahead and proceed with feeding. So I pulled the tube out because I know how to tube feed, and I made the owner pass the tube and then proceed to tube feed herself. So he's making a little bit of noise. You can hear him crying. So we know that he has effectively got the tube in his esophagus and not in his trachea. She's carefully holding onto the tube and slowly putting the formula in. So this is tube feeding 101. Just remember the 5 P's and you can very effectively tube feed puppies. For the people who say that it's not worth saving that little runt puppy, this is him, the same Rottweiler puppy that you saw here. This is him at 9 months of age. And he clearly wasn't a throwaway puppy. He clearly won best puppy in match at 9 months of age. So it is worth doing this. I think it's really important that everybody that raises puppies, especially small breed puppies, are capable of doing tube feeding. I don't believe anybody should starve to death. If you look at my husband, you can see that that's a pretty good philosophy. So no one starves to death under my roof. Everybody should have a chance to survive by at least being fed. There's lots of people that lose puppies for bad reasons, but starvation should never be one of them. When you're starting to wean, you want to make sure that the puppies get adequate amounts of nutrition. You want to start off with a gruel and then slowly move them onto a kibble-based diet. They usually don't stay quite this organized, at least at my house, when they're feeding. I'll start off with a gruel-based diet in a heavy, shallow dish and make sure that all the puppies get up to the dish so that nobody gets left behind. Frequently, you'll find somebody sleeping in the dish. It's a little bit messy when that happens, but that's okay because mom will come in and she's more than happy to lick them and clean them up at that point. Now, different puppies wean at different rates, and this is something we were talking about last night. My farm dog puppies will nurse until they're 6 or 7 weeks old and want nothing to do with eating anything solid. Every one of my farm dog litters have been that way. And I finally ended up with a bitch that was vomiting for her puppies. I was outside in the dark one night and I realized they were nursing and she was like, okay, you guys really have to be done because I'm just going to vomit here and you eat that. My corgi puppies at 2 weeks, they'll start taking gruel. Maybe by 3 weeks, they'll start taking gruel because by 3 weeks, my corgi bitches are like, I'm done. You have teeth. I'm not going to let you nurse anymore. So there are definitely differences breed to breed. in even the smaller breed dogs. I mean, my farm dogs are 20 pounds, my corgis are 24 pounds. There's not a big difference in size, but there's a big difference in attitude. So there are times that you really have to work hard to get these puppies onto solid food because the bitches are just sick of them. And you can't really blame them. If you've put your finger in those mouths, those teeth are little shark teeth. So very important that we get our puppies in front of our nutrition, our bowls. This is kind of a fun little video we took of a puppy at the practice. He was a singleton, kind of a little bratty thing. So just kind of a fun way for us to give him some environmental experience, not that he's going to live on a diet of ginger snaps and peanut butter, but it gave him something different to do while we were trying to do our visit. Water is also a super important nutrient, the most important nutrient. And sometimes we forget about that. We get so wrapped up in getting them to eat that we forget that they need to drink as well. This is a chicken water. The green and white item is a chicken water. It's nice for outside use. It's nice in the house as well. The puppies can't spill it. They can't lay in the dish. If it's outside, they don't end up with all kinds of yucky stuff in the water. And the picture on the right is a Lixit bottle. If you've never used one, they're kind of fun. You can buy them online. They're very easy to find. And I can get my puppies by the time they're 6 weeks old very effectively, and sometimes younger, drinking out of a drinking bottle. This is one that the person just built a PVC stand for so that it's easy, but you can hang it on an X-Pen or a crate. And it's a really great way to get puppies to get some water without having to worry about them going for a swim. Then there was a question that we received prior to this webinar, so we thought we should probably answer this, is when I'm trying to decide if I have a small breed dog between the Purina Sport 3020 or the small breed puppy, which of those diets that I should be feeding, and they're both a suitable option nutritionally. They're both designed to develop to support all stages of growth and reproduction. So they're both fine nutritionally for pregnant dogs, for new puppies, for any of those. The small breed puppy is a little bit higher in protein. The sport has a little bit more glucosamine, EPA, and DHA for joint development, but you're not going to go wrong with either one. The small breed puppy, of course, is a slightly smaller kibble. So if you're not softening the food, you might want to feed the small breed puppy. If you are softening the food, the larger kibble of the Purina Sport 3020 can be softened and then fed after it's mashed down. Just want to remind you that I do have small breed dogs. I do not just have corgis, but I have Danish Swedish farm dogs as well. So I live the same life you do with small dogs. And the dog on the left is running Fast Cat in the pouring rain. This is Dream. And she was absolutely determined that she was going to run when all the other dogs were like, ew, it's wet. This was last September. Oh, I can't run when it's raining. And then the dog on the right is a dog that I sold to a family when they wwere visiting in the desert. This is a picture of him. You can see these are pretty athletic dogs for being small breed dogs. So for dogs that are this athletic, they don't want to eat solid food. They want to just nurse on their mommies as long as possible. So I live the same life you do. So thanks for hanging in there. I want to remind you, we have a book. So if you need anything with that, there's always that to be helped with. And then we're going to talk about the diet a little bit and then take a little break before we do Q&A. Hi, you're back.
I'm here. I'm Bernice Lacey with Nestle Purina Pro Plan. And before our quick break, I just want to take a moment and tell you a few things about Pro Plan. Dr. Greer has already gone over a few of them, but I'll just highlight a few of the items for you. This is what we offer for small dog breeders. We have a lineup of veterinarian recommended small breed formulas with key benefits, including our puppy formula, which also meets the needs of females during reproduction, high in protein to meet the energy needs of high in protein to meet the energy needs of active dogs, small dogs, bite-sized kibble. Real chicken or salmon is always our first ingredient. Natural prebiotic fibers for digestive health. Small breed formulas are good for dogs up to 20 pounds, which is inclusive of the toy breeds as well. And the small breed puppy and the Sport 3020 are both suitable for small breeds. They are higher in protein. The puppy is higher in protein to support the small dog nutrition. Sport 3020 has higher levels of glucosamine, EPA, and DHA to support active dogs. And the small breed puppy has smaller kibble, so that might be the factor when you are trying to decide on a formula for your breed as well. Next slide, please, Dr. Greer. If you're interested in trying Pro Plan, the Pro Club loyalty program for breeders offers great benefits, including savings on every purchase of Pro Plan, free delivery for large orders in most regions of Canada, breeder savings at select retailers, as well as a rebate program for purchases in store. We have a great breeder puppy kits with full bag of free food, so not just the small pouches. This webinar was made by the Canadian team, so the program is specific to Canada, but if you live in the United States, please feel free to reach out and we'll give you some information on the program used in the States. To learn more, you can contact us or sign up at purina.ca/ProClub, or if you opted in to hear from a Pro Plan breeder representative when signing up for this webinar, that would be me. I'll be following up with you over the next few days, so watch out for that email. I've been with Purina for over 18 years, so I'd be happy to answer any questions on Pro Plan that you have. I can also, if I don't have the answers, I'll make sure we have someone that does. So we're going to take a quick 5-minute break. What time is it now? 9:09. So if you want to come back at around 9:15, we'd be happy to answer all your questions. Thank you.
Everybody ready? Dr. Greer, Dr. Westra? There's
Yep.
Dr. Greer. Do we have Dr. Westra? And there he is. All right, let's go then. The first question comes from Terry Fudge. I feed Pro Plan, but add Fresh Pet to make it more interesting to them. Does that work?
Well, I don't really think that it's going to improve your nutritional intake. I would feed the canned Pro Plan and try to stay with that nutritional balance that we have from Purina.
All right, then Karen Wog, I have a bitch who just went into season a few days ago and will be bred this cycle. She's eating Pro Plan salmon and rice. She's already on the higher end of normal weight in preparation for breeding. Is there a benefit to switching her to the Sport 3020? And if so, when should I make that switch?
I think the Sport 3020 is a better diet for that stage of the dog's life. So I would switch her as soon as you can, as soon as you can make that transition over a few days so you don't do a sudden switch. But yes, I would absolutely go ahead and get her switched over, but watch her weight so that she doesn't get too heavy.
Elizabeth Fisher, should you feed grain-free diets?
No.
Quick and easy. Tracy Buck.
Agree.
Is there a recommended guideline regarding size disparity between small dam and larger stud?
Yeah. We were taught in vet school that the bitch will dictate the size of the puppies, and that's not always the case. So, and we do see people that are doing some mixed breed combinations. But even in purebred dogs, you can see some disparity. Clearly, if you're breeding a Bernese Mountain Dog to a Cavalier, you should have the Cavalier be the male and not the female. And yes, that's happening. But I do want to be careful. I probably don't want the male much bigger than about 10 or 20% bigger than the female, just to make sure that she doesn't have ginormous puppies.
And Juhelka writes, how many times a day should a small breed dog be fed? I feed twice a day, but I know other show breeders who only feed once a day.
2. Everybody at my house gets 2 meals a day.
All right. Maureen McCowan, when pups are consistently eating kibble, free feed or put them on a schedule, say 3 or 4 times a day?
It depends on what age they are. If they're really little, I try to pretty much leave it out all the time. But once they get to the point that I'm trying to crate train them and get them into new homes, I will start feeding 4 meals a day, then 3. I think until puppies are 6 months old, they should be fed 3 meals a day because their little tummies are so small, they can't get enough nutrition with 2 meals a day.
Okay. Diane Washer, I have large and small dogs. I'm using the adult sensitive stomach formula for all sizes. Is this sufficient nutrition for the small breeds?
It should be, yeah. I thought there were some DNA questions I saw on here. Where'd they go?
I don't know.
I answered him.
Oh, okay.
You answered them
I
yourself.
thought I was going to get a break.
All right. Okay, we answered that one. Brenda Lawson. Over the course of a pregnancy, are there any guidelines regarding increasing food, or do you just let her guide you as per her demand?
That's a great question. You should, from the time that you diagnose the pregnancy at 4 to 5 weeks, then we recommend increasing the food intake by 10% a week so that she's consistently getting increased amount of caloric intake during that time period. I did not mention that, and I should have. Sorry about that.
All right. Diane Washer, my Cavalier is pregnant. I started feeding her the sensitive stomach puppy formula. Is this sufficient? I have also added a quarter tablet of folic acid 2 times a day. Is this okay?
I don't know how much folic acid is in your tablet, so it might not be enough. And the sensitive skin and stomach diets are absolutely fine for the dogs nutritionally. They don't have some of the real fine details that I think we probably would prefer with the sport diet. But if you have a dog with GI or skin sensitivities, then the sensitive skin and stomach is going to be a better choice.
All right, Carol Brunstein wrote, what folic acid supplement do you recommend if the kibble being fed doesn't have it? Or how do you know how much folic acid to feed?
Sure. And there's folic acid in pretty much every diet. The question is, how much is there? The only published dose we have on folic acid is 5 milligrams per dog per day. And most of the folic acid supplements are 400 micrograms. That means 12 a day for a dog, which is ridiculous. Like nobody's going to feed their dog 12 capsules a day. So we don't have a better published dose. On a small breed dog, I would probably do 2 of those a day, but I would really like to see some research on this.
Debbie Coate wrote, I feed a totally prepared raw diet. I've tried to switch them back to kibble, but the dietary issues, runny poops and vomiting has made me switch back to raw. How can I make this switch easier on my Yorkies?
A really gradual change. Probiotics to help, because anytime you change the diet, it can take up to 2 weeks for the GI tract to adjust to the newer type of food that's in there. I'm just really not big on raw during this time period. I think it's really important. So I think we just very carefully have to make that transition. I used to have cats that we were breeding, and the same thing happened with us with those. Is we had the breeder that was feeding them a raw diet, and it took a little time to make that transition. But once they got back to a kibble-based diet, it worked out fine.
Corey Zimmerman, is it okay to feed the bitch after having puppy kibble mixed with boiled chicken, brown rice, and boiled eggs? She's a 10-pound female.
Yeah, and sometimes they just don't eat well when the puppies are really young or right after they've gone through a pregnancy and whelping. They may have eaten all the placentas and then their little tummies are full of placenta instead of dog food. I try to say not to exceed 10% of the dietary intake with food that isn't commercially produced dog food. But there are sometimes just a few days in that time period that you have to get something into them. So brown rice, boiled eggs, and chicken is better than no food. But I would try to make the transition back to a kibble as soon as I could, or canned food.
Okay. Lori Quenville, I have labs and use the 5 milligrams, and I'm hoping to breed Frenchies. I wonder if it was the same amount.
I would on Frenchies because they're so prone to cleft palate. So I'm assuming she means folic acid.
Okay. Debbie Evans, I have Cavaliers. I'm finding it hard to find a cardiac vet to do OFA testing. What can I do to make my puppy owners less fearful? Are there tests that my vet can do to help? I've done the DNA testing, but not genetic testing.
Dr. West.
So one thing is like when we talk about DNA, a lot of times that's just profiling in parentage through AKC. So it's very distinct from doing the genetic test for the known disease-causing mutations. You are correct. Unfortunately, cardiac disease falls into that polygenetic category. And so there are a limited number of genetic tests available, none of which are really good at kind of screening your pets. for the risk that they may produce. So you are left with doing the phenotypic test, which means having your dog echoed by a qualified veterinarian to look and evaluate the heart. And again, this kind of comes down to what we call conservative breeding strategy, which means, if we breed good to good, we increase the chances of getting more good. And that's unfortunately where we're at. The difficulty is, especially with the mutations that we do understand about cardiac disease, is that we know that they can cause significant disease in some breeds, and yet other breeds can be fully, carriers or, basically have two copies of the mutation, and yet we don't see the mutation, which again speaks to our limited understanding of gene expression. So kind of left with going with the cardiac exam is your best bet.
Caitlin Vance, calcium supplementation for lactating bitches. I have frequently heard they cannot be overdosed on calcium by oral means. Is this true?
Yeah, it's really hard to overdo it. Now, there is a practical limit, whether you're using calcium tablets that are 500 or 600 milligram, or you're using Tums or whatever. It's pretty hard because the GI tract will only allow a certain amount to be absorbed, but you want to be reasonable. You don't want to give an entire bottle of Tums to a small breed dog. So be practical about it, but it is pretty hard to do it by oral route. You can give too much calcium if you're doing it by injection, but orally do what seems reasonable. 500 to 600 milligrams, 2 of those a day for a large breed dog, 1 of those a day for a small breed dog is plenty.
Elizabeth Fisher, Dr. Westra, if your line, dandies, have glaucoma, great-great-grandfather, should you be overly concerned about this popping up, especially if you have your bitch's eyes tested yearly and her offspring?
You're doing it right. So with glaucoma is that there are some genetic foundation for the presentation of the disease. And interestingly enough, the mutations are dominant, but there's a mutation associated with disease in terriers, and there's a mutation associated with disease in border collies or Australian shepherds. And so you're looking at a very, kind of almost a breed-specific mutation, and at the same time, there's a large component of other etiologies that will produce glaucoma. So if you know that it is somewhere, in the line, your best bet is to have your dog's eyes checked on a regular basis, just because, again, genetics only knows, enough to be dangerous.
Okay, Maureen McCowan, is it harmful to feed whole egg, especially during pregnancy and whelping period? Mine steal right out of the coop.
Well, too many raw eggs can lead to a biotin deficiency. So if you can talk the dog into having them boiled before they eat them, that would be more convenient. But I'm pretty sure with your description here that that's not going to happen. Yeah, there's concerns with that. Also, salmonella and some other things would be reason for concern. So I don't know. I think you're going to have to do a better job fencing the coop off so that the dog doesn't continue to do that, because those homegrown eggs are always the best. I don't know why the dogs are stealing them from you. They should be saving them for you.
Shannon Miller, please repeat your answer. How much folic acid to give? Tablets or 400 milligrams? Thank you.
5 milligrams is the only published dose, which is if you have 400 microgram capsules or little gel capsules, it's 12 of those a day. That's not practical for small breed dogs. So typically I would do 2 400 microgram capsules a day. It's a water soluble vitamin. So you're not going to overdose on it. It's just that there's going to be excessive amounts that go out in the urine if you give too much. So it's not going to hurt anything. It's just the amount of money that you spend.
Debbie Cote writes, we had a pup with a liver shunt. Both parents tested clear, but I understand that there is no genetic test for something defined as polygenetic. Should I remove this breeding pair from our kennel or just not breed this pair again?
So you're touching right exactly on how we kind of approach those conditions. So if we assume that the condition is recessive, and we make that assumption based upon the presentation in the litter. So if you bred the parents and all the puppies had liver shunts, then you would probably suspect one or both of them are carrying a dominant mutation. But if you bred the parents and you had one affected puppy, then if there is a genetic component to this, it's likely recessive. And so the way we kind of skate away from that is that we don't have to retire the parents, we just don't want to put them back together again. Now, there's another half of this, which is a little bit of a challenge, and that is if you're looking for a new potential mate for one of those pair that you're trying to produce, it's a good idea to have good, solid communication with whomever you're looking to, either get semen from or to talk about who you're going to pair that dog to, because you find out if they were ever a part of a pedigree that produced a liver shunt. And it's imperfect, but that kind of helps kind of increase the chances that the pedigree that you're now kind of reintroducing the dog to is actually free of that mutation. It's not recessive and hidden somewhere in their line.
Patty St. Cyr, how would you rate prepared foods like farmers' dogs versus the usual kilo prepared like Pro Plan or Science Diet, etc.?
Sure. So the Pro Plan, Science Diet, and Roakin and Eukanuba Waltham, those, to my knowledge, are the only three companies that have large-scale testing of their food, not just on a chemical basis, but in feeding trials. So I would contact Farmer's Dog. They should have an 800 number and ask them what their feeding trials, how many dogs are in their feeding trials, what ages of the dogs, what breeds do they use. I would really drill down on that. And see what they can tell you. The World Small Animal Veterinary Association, WSAVA, has a great website that has questions that you can ask when you call the pet food companies and ask them what they do for feeding trials, ask them how they make the determination. Do they have a nutritionist on staff? They have a great series of questions that I'm not going to repeat because I can't remember them all. But WSAVA, World Small Animal Veterinary Association, you don't have to be a vet to get on that website. So look it up, pull up the list of questions and call the company and ask them. And then call Purina and ask the same questions and see if you like the answers.
Perret Kekonovich, Dr. Westra, do you have a recommended books on genetics for breeders?
Well, that's a really good question. I think I should employ Dr. Greer to write one.
Gerald Bell has a great book, Veterinary Medicine for Dogs and Cats. It has a great, it's like 500 pages. It's a great book. The old one Ross Clark used to have is really, 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 really old and out of date. But honestly, almost any book is not going to be in date because DNA tests are coming to market every 15 minutes. So there's not going to be anything in print that's completely current. You're going to really have to count on websites for that, don't you think?
Yeah, that's exactly what I was going to say, is that a lot of the texts are almost out of date now. Some of the things, I think the advantage of getting just, a general text is just understanding how to use or employ genetics, because I think, that would have just as much value. If you're looking at what's new and what's available, I think, you're going to have to get out there and kind of look at the different companies and what they're offering. And then, of course, reach out and, that's one thing is that we're happy to discuss any questions or concerns you'd have about the availability of any tests.
Oh, I see Alice put the link for WSAVA in the chat. That's brilliant. Thank you, Alice.
Caitlin Vance, Dr. Westra, I have mini bull terriers and we are dealing with size creep as we have dealt with several health issues and type. Do you anticipate a near future where the DNA tests for size size will reasonably help us reduce size?
So that kind of goes to that one slide that I kind of breezed through a little bit, was that because we're dealing with, only limited ability to test, and then with the variable expression that you see in that, I want to say that it's going to be getting better, especially. So I'm going to jump into this, is that sequencing is going to become much more commonplace in genetic testing. Currently, right now, we use point mutation, which we're looking for very specific genetic markers to identify the genes that we're testing for. But as we start to look at sequencing, which means we're looking at whole swaths of the genome, the polygenetic tests are going to start being produced. And just to kind of explain it, is the first thing that's going to come out there, they're all going to be risk assessments, which means that you're going to see things come back as high risk, medium risk, unknown risk, things like that, which will be a challenge initially when it comes to interpretation. But I think that, in the not too distant future, these risk assessments are going to become much, much more commonplace. And they're going to start to become much more functional for the breeder to get away from some of these concerns.
Liz Braun, is it common for juvenile cataracts to skip generations? My male has a brother from a later litter affected, and looking back in the pedigree, there are other dogs that presented with cataracts. My dog has been served in the last 3 years, and I plan on testing his puppies this fall from his first litter.
So there's one mutation that we know of that's dominant and one that's recessive. So when we talk about skipping generations and they come from a genetic etiology, then we're dealing with the recessive, and that is effectively a carrier, was just kind of continued through the pedigree until it met another carrier, and that produced the effect. And suddenly it's like, boom, there it is again. Where did that happen? The thing about cataracts, though, is again, there are other ways a dog can present with cataracts. So not all cataracts are genetic, which can be frustrating.
And Faust, if you have a bitch with grade 1 luxation intermittently, should you mate them? If so, what risk are we taking at producing puppies with that issue?
A great question. Again, it's a big concern, especially with our toy breeds. Again, it comes down to breeding like to like, and with the increased, with the hope that we're going to produce more like. So it's almost impossible, because when we're talking about risk, we're talking about relative risk, and what we really want to know is absolute risk. Like, what is the chances, or what is the puppy going to result with from that pairing? And there's just no good way of doing it right now. Again, when it comes to luxating patellas too, especially if they're just grade 1, you need to don't lose sight of the trees through the forest, if you will. That means that, if you're focusing just in on the patellas, you could be missing out on some other positive feature or trait that dog could conserve in your program.
Christine Tremblay, how is polydactyl passed down, and how should we approach breeding dogs that have produced 1 pup out of 6 in the litter?
So the mutation that we test for polydactyly is very breed specific. And so if you're seeing it result in other breeds not associated with Samoyeds and Great Pyrenees, it's likely what we call a de novo mutation, which means that it is just something happened in that dog, which produced that result. Now, again, I hate to sound like a broken record. We don't understand all the presentations of mutation, but for the ones that we see, a lot of times is that they can have that condition, and it's not what we would call inheritable, which means that it's not likely to be passed on.
Randy Black, Dr. Westra, will you have a DNA test for cleft palate markers, assuming that would even be a thing?
We do. It's for the Nova Scotia duck-toling retriever, and it is for it's for a syndromic presentation of cleft palate. And so, as Dr. Greer talked about, there are other ways that cleft palates arise. The unfortunate thing here is that oftentimes the genetics involved in the production of a cleft palate have multiple effects throughout the embryonic development. And so usually the mutations that we understand usually lead to almost kind of disaster presentations. So it hopefully was something that we'll be able to better identify here in the near future. It's something that's actively being researched, and that's why we have the one that we do. So the answer is quite possibly.
Very good. Okay, everyone. I think that's going to be it for tonight for the questions. It's been a great session. Thank you so much, Dr. Greer and Dr. Westra, and to everyone for joining us and participating, and to Pro Plan and Paw Print Genetics by Neogen for organizing this session. Thank you so much, everybody, and have a great night.
Great questions. Thanks, everybody.
Good night.
Thanks, doctors. That was great.